Next speaker is Stavrini Papadaki from Greece.
Hi. Hello, everyone.
Yeah, she is working in China, right? West Lake University.
Yes, exactly.
With Kirill Pyatkevich. And so everything was mixed up. China is completely different time zone, but
oh yes, good morning.
but
good night, actually. I'm in Greece.
Oh no!
Okay, please go ahead and present your work, Stavrini.
Okay, let me share my screen.
Dual expression vector for blue fluorescent protein optimization.
Can you see the screen?
Yes, we do see it.
Okay. So give me 1 second. Okay, so thank you very much for giving me this opportunity to present part of our work. Today I will be presenting our recent work on the development of a dual expression system for blue fluorescent protein optimization. I will start with a brief introduction exploring part of fluorescent protein applications, as well as introducing one of the main goals of our lab. Following will be the theoretical basis of this project, where we will briefly discuss about the chromophore maturation pathway and the approach we use to develop 2 new blue fluorescent proteins. Subsequently, we'll move on to the performance of the 2 blue fluorescent proteins named Electra-1 and Electra-2 in cultured mammalian cells and in vivo in a side-by-side comparison with top-performing BFPs. We will also discuss our strategy to further improve Electras to better suit our needs, and finally our conclusions from this study. Now let's start with why do we need another fluorescent protein? One of our lab's major goals is to use multiplex imaging to facilitate neuronal tracing. In order to achieve this, we need spectrally resolved, bright, photostable, monomeric, and chemically stable fluorescent proteins. Now, there are already many different strategies to implement for multiplex imaging, and as an example, I have here the split FPs or the magic marker strategy. With each of these existing strategies can be combined with expansion microscopy protocols in order to increase resolution and overcome the diffraction limit. But the more fluorescent proteins we have with the aforementioned characteristics and properties, the richest our color palette will be. Now, why do we need another blue fluorescent protein? Because blue fluorescent proteins are characterized by lower molecular brightness compared to green, yellow, and red fluorescent proteins, as you can see here in this table. One implication, though, to keep in mind is that molecular brightness can differ greatly from intracellular or in vivo brightness. And this has been observed in several studies, and we believe that it also creates a need for testing newly developed and also existing fluorescent proteins under different conditions and possibly in various expression systems. In this study, for example, 7 fluorescent proteins were selected and compared based on the reported molecular brightness. As we can see here, for example, M-neon green has the highest molecular brightness, but when expressed in C. elegans embryos, it has the lowest molecular brightness compared to GFP and M. whitehead. And same goes for M-rabi2, actually. These results increase the need for thorough testing of fluorescent proteins, both for those that develop, but also for those that choose fluorescent proteins to better suit their experimental needs. Keeping these in mind, let's move on to the theoretical basis of this study, which involves the DS-RED chromophore maturation pathway. Now, in order to convert a red fluorescent protein into a blue, we need basically to block the maturation pathway in the blue precursor, while in parallel we stabilize the blue form of the chromophore. In our case, this was achieved by mutating a few key amino acid positions, both in the chromophore as well as in the chromophore's environment. And our main goal was to prevent oxidation and transition towards the red form, as well as degradation and transition into a non-fluorescent form. The experimental approach that we used was called hybrid evolution, and it combines directed molecular evolutions, as was described before, with hierarchical screening, meaning screening both in bacterial and mammalian cells using the same expression vector. Before we move on to the development of the blue fluorescent proteins and their performance, let me introduce our basic tool in this study, the p-hybrid vector. As is shown in this map, p-hybrid combines both bacterial and mammalian expression cassettes for the target protein, in our case the blue fluorescent proteins, with mammalian expression cassettes for the reference protein, where we use M. scarlet. To validate this vector, we cloned 3 BFPs in the target protein position, keeping M. scarlet in the reference position. As you can see, all 3 target proteins were successfully expressed in both systems, bacterial and mammalian cell culture, while the reference protein was only expressed in mammalian cells. This vector allows the hierarchical screening of variants in bacterial and mammalian cultures, but its great advantage is that it eliminates the need for recloning, which is usually the most time-consuming step. Our experimental design can be summarized in this slide. So first, we selected our template, a red fluorescent protein, mRAB3, which is among the top 3 fluorescent proteins with the highest molecular brightness. This template was subjected to directed evolution by mutating key amino acid positions. The mutants were cloned into p-hybrid vector, and E. coli cells expressing single copies were sorted using FACS for exclusive blue fluorescence. From this library, the brightest colonies were selected for protein purification and in vitro characterization, as well as screening in mammalian cell culture. The top-performing variants were subjected to 2 more rounds of random mutagenesis, following the same process and selecting basically for enhanced properties. After 2 rounds of random mutagenesis, the 7 best-performing variants were selected for a final round of side-by-side comparison, which resulted in Electra 1 and Electra 2. Moving on to the results, here you can see the final comparison of the 7 variants in terms of brightness and photostability, as well as the excitation and emission spectra of these 2 proteins. Now, our first goal was to compare their performance side by side with top-performing blue fluorescent proteins in cultured mammalian cells, starting with HEK cells. As is shown here, both Electras outperformed M. blueberry 2 and EBFP2 in terms of brightness, but they were significantly dimmer compared to MTAG-BFP2. During photostability measurements under high-power illumination, we observed a somewhat different behavior, photobleaching behavior, compared to the one we saw during low-power imaging of brightness. So we were prompted to also measure their photostability under low power. And as you can see, there are some really quite obvious differences, especially for M-Blueberry 2. The next step was to test their performance in cultured primary mouse neurons. And while the ranking in brightness is very similar to that observed in HEK cells, photostability behavior is completely different, with eBFP2 being the most photostable and MTAG-BFP2 being the least photostable. Already now in these results, we observe different performance as we discussed in the introduction, but this difference becomes more apparent in the in vivo applications. Before moving on to the in vivo applications, we wanted to test Electra's monomeric state. For that, we used the Osser assay, where the proteins were fused to the cytoplasmic end of an ER membrane protein. This fusion leads to a transformation of the ER tubular network, creating organized, smooth ER whorl structures, as the one you can see here. Now, the number of cells with whorl structures, as well as their brightness, has been correlated with the monomeric state of the protein. Low brightness, for example, was correlated with low tendency for oligomerization. Additionally, high number of cells with normal ER was also correlated to low tendency for oligomerization. In our hands, both numbers for Electra 1 and Electra 2 were very similar to MTAG-BFP2, a monomeric blue fluorescent protein. But since the percentage of normal cells is quite low, and this is a normalized to 100 number, we decided to test both Electras, how they perform in demanding fusions with structural proteins. As you can see in these representative cells, the Electras show overall correct localization, leading us to conclude that they probably have low tendency for oligomerization. To further explore the utility of Electras for multicolor neuroimaging, we expressed them in vivo in model organisms, including C. elegans, zebrafish, and mice, using MTAG-BFP2 as a standard for comparison. In all three organisms, the proteins were expressed in neurons with Electras outperforming MTAG-BFP2 in C. elegans in terms of brightness. In zebrafish, Electra 2 was significantly brighter than MTAG-BFP2 when expressed in zebrafish hindbrain, but not in spinal cord, while MTAG-BFP2 was the most photostable among the three proteins compared. Electras were also expressed in the mouse cortex and imaged under two-photon excitation, where they showed even cytoplasmic labeling, with Electra 1 presenting significantly higher overall brightness, specifically in deeper cortex layers. Now, if you recall the cell culture results, you'll see a different performance trend, especially that of MTAG-BFP2, which outperformed both Electras. Here, in the in vivo applications, we see a different pattern. These observations led us to agree with previous studies supporting the validation of fluorescent proteins in various expression systems and using different imaging modalities depending on the intended use. As I mentioned in the beginning, our final goal is to develop spectrally resolved, bright, photostable, monomeric, and chemically stable fluorescent proteins to use them in neuronal tracing by implementing expansion microscopy protocols. Now, one common thing among the EXM protocols is that they all have one initial step of fixation. which is usually a very harsh chemical process, especially for fluorescent proteins. Now, when we compare the brightness of electrons with eBFP2 and MTAC-BFP2 in fixed mouse brain slices, this is what we got. So we were also prompted to test and to measure their chemical stability by basically using the purified protein in a neutral pH and measuring the brightness in a course of 1 week. Our results clearly indicated a very low chemical stability compared to MTAC-BFP2. For this reason, we decided to further optimize their chemical stability. We used the known crystal structure of M-CRISPR, which has high sequence homology with the electrons, and identified possible key amino acid residues in close proximity of the chromophore that could stabilize the structure. Now, to sum up this presentation, we developed a dual expression system for blue fluorescent protein optimization named bHybrid. As we saw in the results, electrodes show differential performance depending on the expression system. When tested in vivo, electrodes proved to be suitable for multicolor neuroimaging. And as a final conclusion, I would add that new fluorescent proteins should be tested in different systems under multiple conditions as their performance may vary and in order to provide valuable insight to the end user. Thank you for your attention and a warm thank you to all the members of our group, as well as our co-authors and our collaborators for this work.
Thank you so much, Stavrini. Questions? Brad?
Do you know why the blueberry FP bleaches faster at low light intensity than high?
Sorry, could you please repeat the question?
So your blueberry bleaching under high and low light intensity.
Okay, so what we actually did not further test why this is happening. We can just hypothesize that probably under high power illumination, some of the protein molecules may not be completely in the mature chromophore state. So given the energy, they are activated, while under low power illumination, assuming this happens, I would suspect that there are no more molecules activated, but rather those that exist are bleached.
Okay, so then it's very nice, and I'm loving this, but it also scares me a little where you show different fluorescent levels in different cell types. And I'm wondering if you looked at the bleaching of those individuals in the different cell types also. Yeah.
Okay. Okay. So we only measured the photobleaching in zebrafish, and actually this data come from the hindbrain and not the spinal cord.
What if you did the spinal cord? Would it look the same or would it be different?
Sorry, I have a trouble hearing.
If you did the bleaching in the spinal cord, would it be similar or would it be different?
In this case, I would expect it to be similar. So usually, at least in my experience, when a fluorescent protein is rather bright, it might bleach very faster compared to other dimmer proteins. In this case, I would expect not a very big difference actually. between hindbrain and spinal cord. But for C. elegans, for example, we had some troubles actually measuring the photostability because the worms started waking up after a while, so it wasn't very easy.
Okay, more questions? No. Thank you, Stavrini.
Thank you very much.
Yeah, can you stop sharing? Stop sharing. Yeah, you did already.
Yeah.
Okay. So we will have a break, lunch break.